Yes, so I'm not a pharmacologist. I'm actually a computer modeler, and I'll be showing you not just how to use in silico models to predict the risk of torsade de pois in cells, or in the heart, I should say. I'll be showing you how to use in silico models to, or how to reduce in silico models down to a very simple metric called the axis of arrhythmia, which you can calculate with a pen and paper. So you don't actually need to be running full-blown in silico models in your own wet lab. Now, I'm sure you know better than me that torsade de pois is a potentially lethal ventricular arrhythmia, and that it can be induced by many different classes of drugs. These drugs don't necessarily target the heart purposely, but you can get torsades from all sorts of drugs, including antihistamines, antibiotics, antipsychotics, and so on. And of course, this is the reason we do safety pharmacology. Now, back in the mid-90s or so, it was discovered that all of the known proarrhythmic drugs at the time were blocking one particular ion channel called the hERG channel. And the hERG channel carries the rapid delayed rectifier potassium current, which we'll just call IKR from now on. Now, IKR is responsible for the repolarization phase of the action potential. So it's responsible for bringing the action potential back down to the resting membrane. So if you block IKR, you block repolarization, and that action potential prolongs. If that action potential prolongs too much, you risk having these spontaneous early after depolarizations in the cardiomyocyte. And these early after depolarizations are thought to be the trigger of the arrhythmia. They cause, it causes reentrant circuits to occur, which then degrade into fibrillation, which stop the heart beating. Sorry, they don't stop the heart beating. They stop the heart pumping blood effectively. It's just sitting there quivering. Now, with the discovery of the importance of the hERG channel, the response was basically the standard hERG test, where we test any new drug for hERG block, and if hERG block is found, then that drug is deemed to be dangerous for torsades, that is. Now, there's a fallacy in that reasoning. The fallacy is the fallacy of asserting the consequent. And that is because all dangerous drugs that block hERG, you have the observation that all dangerous drugs block hERG. It does not imply that all drugs that block hERG are dangerous. And here's an example of just that. Here's a case of a drug which blocks 4 ion channels in this case. One of them is hERG, so it would fail the hERG test, but the way it blocks these ion channels balances the inward and outward currents in such a way that you do not get the early after depolarization in the action potential. You get a little bit of prolongation, but you don't get that early after depolarization. So this drug would be a safe drug, and it would fail, but it would fail the hERG test. So this would be a false positive from the standard hERG test. And this has been known for a while. Since 2012 or 2013, the SIPR initiative was emphasizing the need to look at multi-channel assays. So we're looking at multiple ion currents to try and predict the torsadogenic properties of drugs. And they suggested multiple ways of doing this, and one of that is in silico modeling, which is what we have done. And so we model the membrane of the cardiomyocyte. In our case, we use the O'Hara-Rudy model, but that's quite a complicated model. I won't go into all the details, but in all models, you can consider that the ion channels are represented by these variable resistors. And you can mimic the effect of a drug or drug block on that channel by tweaking the resistance of each of these ion channels in the equivalent circuit. And this circuit will generate simulated action potentials, and we classify our action potentials as either benign or ectopic, according to whether they have an early after depolarization. Okay. Now, ordinarily, most modelers would do that by applying the blocks to these ion channels that represent a particular drug. But paradoxically, we do not. We do quite the opposite. We actually just randomly assign variables to these action potential. We randomly assign conductance values to these ion channels and see what action potentials we get. And we plot out in a parameter map. So here we have the conductances of two ion channels, GKR and GCal. Whenever you see a G here, that means conductance. So this is the conductances of two ion channels, and each point in this map represents one cell. And if the cell is ectopic, it has EADs, we color it red. If it's not ectopic, if it's benign, we color it white. And you can see there's a very strong relationship, a very clear relationship between the ectopic and the benign cells here in this map. Now, please remember that the true map is actually four-dimensional. I'm just showing you a two-dimensional slice here because it's easier to visualize. Notice also that we can fit this dashed line to that boundary between the red and the white. And we can do that using linear statistics. And once we have that boundary, which approximates the behavior of the cardiomyocytes, then we can begin our analysis. So take 2 points P and Q on this map. They represent two different cells. The difference, or point P, is a normal cell. It has a normal action potential, whereas point Q is quite abnormal. It just has this train of early after depolarizations. Now, the only difference between P and Q and the way they're configured is the conductance of the two ion channels here. And the difference between those conductances can be equivalently interpreted as a degree of drug block. So you can think of P and Q as two different cells, or you can think as P as the cell before the drug. You apply the drug to P, and you have this Jekyll and Hyde effect where Q is the drugged cell. So now we can analyze how that drug affects P and turns it into Q, if you like. And in this case, this particular drug sends the cell into this pink region where we have ectopic beats in the cell, and so we call that a pro-arrhythmic drug. Now, you could have two drugs, in this case, drugs A and B. So in this case, drug B shifts the physiology of the cell from P to R. So R is a benign action potential because there's no early after depolarization. It is a little bit prolonged, but it's still normal in our classification system. You might notice that both drugs A and B have the same degree of block on GKR. So that's equivalent to the Hurg block. It's like they both have the same Hurg block, but we have two different outcomes. One pro-arrhythmic, one benign. And we can extend this reasoning and generalize it so that any drug which takes your system or your cell P towards that pink ectopic region is classified as a pro-arrhythmic drug. Any drug which takes that cell away is an anti-arrhythmic drug. Any drug which moves you parallel to that dashed boundary between the pink and the white is a neutral drug because it has no impact on torsades, or at least has no impact on early after depolarizations. And that gives us the axis of arrhythmia. So the axis of arrhythmia represents the shortest pathway from anywhere in the white region towards the red region. So it's the shortest pathway to go to force a cell to be arrhythmic, or arrhythmogenic. Okay. Now you can think of, I should say, it is defined as being perpendicular to the dashed line here, the decision boundary between the pink and the white. So you can conceive of the axis of arrhythmia as representing the worst drug that is theoretically possible for generating torsades in a cardiomyocyte. Now, I know the worst drug's not the best name, but it gets the idea across. It's not particularly scholarly, but it gets the idea across that it's a conceptual drug and it's the worst drug you could possibly have. There is no other drug that can be worse because of because we're assuming linearity here. But once you have that worst drug defined, you can then use that as a yardstick for comparing to real drugs. So here's a real drug A. It blocks the currents in such a way, and you can compare drug A to B by projecting the vector A onto the vector B. And the length of that projection is the risk of torsades that drug A has. The other way to think of it, it's the component of drug A that matches the worst possible drug. And that the length of that risk vector is easily computed using dot products, which is something you might remember from high school or perhaps undergraduate mathematics. A dot B is the projection of A onto B. Now, some properties of this risk score. If you have a positive risk score, it means the drug's taking you along this line towards the danger zone, I'll call it. And if you have a negative risk score, it's taking you away. The neutral risk score is 0. So here's some examples. This is ibutalide, which is a pro-arrhythmic drug. It's a potent IKR blocker. Funnily enough, it's actually used to terminate atrial fibrillation because it lengthens or prolongs the action potentials in the atrium, but it also prolongs the action potentials in the ventricles, and that's where it causes torsades. So if we plot the drug action vector for ibutalide here in our parameter map, project it onto our axis of arrhythmia, we get a risk score of 1.44, which is a positive risk score, meaning there's a positive risk of arrhythmia. Here's a counterexample. This is linezolid. Linezolid is an antibiotic and a potent calcium blocker. It has this drug action vector, and when we project that onto the axis of arrhythmia, we get a minus risk score, suggesting that this drug is indeed safe for torsades. Now we can, those drugs were, those two examples I gave you were fixed at 10 times the therapeutic dose. When you increase the doses of those arrows or those drug vectors do actually change shape. They have a curvy linear trajectory. So here, if we look at the left, I'm increasing the dose of the drug, and you can see the trajectories. Sometimes they curve, sometimes they don't. It depends on whether one ion channel saturates before another. And the projection of these vectors onto the axis of arrhythmia is plotted on the right. And so we have these risk scores that vary with dose. And here, any of these risk scores that are above 0, we classify the drug as unsafe. And any risk scores that are below 0, we classify the drug as benign. Now, we have a data set, we have access to it's a publicly available data set of 109 drugs from Lopus Lorete, which have clinical risk labels from Credible Meds. So we can plot these trajectories of the 100 drugs, compute the risk scores. Here, the colors correspond to the clinical labels. So unsafe drugs are in red with the clinical labels, and the clinically benign drugs are in black. And we compare the color codes of the risk labels with the predictions from the the risk score that I've computed, and we get an accuracy of 89.5% plus or minus 1.4. There's a bit of variation because it depends what dose you actually assess these drugs at. But what I want you to notice at this point, actually, is if you're eagle-eyed and you know some statistics, if you have a look at the parameter map on the left, you might see that you could actually draw a decision boundary right through those drug trajectories. And you can separate the red and the gray trajectories just using a linear boundary there without having to do any of those computer models. Just by using statistical analysis of these drug trajectories, we can estimate this decision boundary. And in theory, this decision boundary should be perpendicular to the axis of arrhythmia. So now we have two ways to estimate the axis of arrhythmia. The first way was using the computer models of early after depolarizations. And the second way is just using straight statistical analysis of those data sets. And that's what this diagram shows here. So the first way I showed you was the top. We get the axis of arrhythmia estimated from the computer models. And the bottom way is through straight statistical analysis of those drug block data sets. So this is good. We have two quite independent ways to estimate the same thing. And so we do that. These bars on the left represent the coefficients of the axis of arrhythmia. The coefficients are just the slopes, basically, in each dimension. Remember, there's four ion channels, so we have 4 coefficients of that axis. The blue are the estimates from the drugs, and the red are the estimates from the computer simulation. So you'll notice that the axis coefficient for calcium from both methods are very consistent, as are the estimates for BKR. Not so much for late sodium. So BNAL, we have quite a discrepancy between the estimates from the drugs and the computer simulations, and a really big discrepancy between the estimates for the drugs and the data simulation. Estimates between the drugs and the computer simulations for BKS. In fact, they go in opposite directions. So this causes us a bit of a problem. The computer simulations and the drug data are in disagreement, and we don't know which is true. Both have their problems. So what do? We just don't use them. We just throw away those two problematic ion channels. We say, we don't know whether the model's wrong or the data's wrong or a bit of both. Let's just stick with those 2 ion channels. So L-type calcium and the Herd channel, and we'll just use those 2 channels only. And when we rerun the whole process, we estimate those parameters of the axis of arrhythmia again, and we find really good agreement between both methods. Really good agreement. By really good agreement, I mean there's no statistical difference between these 2 bars, the red and the blue bars. And that's quite amazing because they are really different. They come from really different sources, this information. Remember, we're comparing a computer model that generates early after depolarizations versus a straight out data analysis of drug block based on clinical labels with no biophysical modeling at all. Yet they both give almost identical estimates of the axis of arrhythmia. So that convergence, I think, is quite amazing. So how does it perform? I should say, so we take both of these estimates and we pool them together, and that gives us a pooled estimate of the axis of arrhythmia. That's the black heavy line there. The gray region is the 95% confidence interval. And this is our simple metric of Torsard's risk using that axis of arrhythmia. So all you have to do is take the alphas and the betas, multiply them together and add them up. I give you the betas. The betas are the slopes of the axis of arrhythmia. Remember, that represents the worst drug. And you compute the alphas yourself from the fractional conductances of the drugs that you've developed. And you already know how to do that. The alpha is simply the logarithm of the fractional conductance. So you can do this already, and it's just a simple matter of calculating those risk scores based on the various concentrations of the drug that you measure in your patch clamp machine. And we've done this, we've computed these risk scores for those 109 drugs. And again, we get 90% accuracy, 90.8% accuracy. So we've gained 1% of accuracy above what we had with the 4 ion channels. But how does that compare to the standard Herg test? So here I'm comparing the results of the axis of arrhythmia on the right with the standard Herg test, which is the 30-fold Herg safety margin. So you probably know that the Herg safety margin here is if you take 30-fold or 30 times your therapeutic dose, if that still is below the IC50 of Herg, then you're within the safety margin for Herg. And if we use that criteria for predicting whether the drug is safe or not, we get 86% accuracy compared to the clinical labels for those known test drugs. Now that compares to 90% accuracy for the axis of arrhythmia. Now more so, we get about a 30% reduction in the false positives. So remember, the false positives were the problem with the problem with the Herg test because of that fallacy of asserting the consequent, and it was the false positives that spawned the SIPA initiative to try and reduce the cost of wasted drugs. And so now we have about a 30% reduction in those false positives using the axis of arrhythmia. Now, the benefits, so we're not just talking accuracy here, but there are some practical benefits of this method. The first is it's an extremely simple calculation to do. You're not burdened by running drug-specific computer simulations. You don't need to run digital twins or anything like that. We just compute this risk score directly from the drug block that you see in the patch clamp experiments. You don't need to know your therapeutic dose, so you can conduct this experiment very early on in the drug development timeline. The therapeutic dose is not part of the risk score. You can compute the risk at various doses, and you can compare those doses to the therapeutic dose later, but you don't need them to compute the risk score. In fact, you don't even need to know the IC50s. In fact, you can just compute the risk score directly from those fractional conductances as they come right off the machine. And because of this simplicity, it is possible. We're currently working with Nadian to see if we can embed this risk score within their SynchroPatch machines to give you these very early risk predictions as you measure the drug block in those 2 ion channels for the L-type calcium and Herg block as you go along. Now, this is still a work in progress. We haven't got this up and running yet, but we are heading in that direction. And when I say we, I should include my collaborators here at the Victor Chang, Jamie Vandenberg, and Adam Hill, who have been developing this with me, and I think we can hand over for questions now, Marta. And we flip over. How are we?
Perfect. Thanks, Al. Thanks, first of all. It was a really great talk. Thanks for sharing. And thanks also for being so perfectly on time to watch. So I, of course, ask the audience to write in the Q&A if they have any question. I myself would like to start. Do you think that then in the future, this axis of arrhythmia that now you identified will become, let's say, specific for patients? So for each patient, that so history of pre-existing condition or genetic specificity?
Well, we are working at the moment from what you might call the average heart cell. Now, the risk, if you recall it, the risk describes how far you move towards that that danger zone, I'll call it, if you can see it here in the pink.
Mm.
Now, it actually matters where you start. So if you are a totally average heart cell, you'll start bang on the origin in this particular parameter map. But you can imagine a cloud or a Gaussian distribution or normal distribution of so it surrounds this origin, which represents the human population. So the risk of the drug actually shifts the entire cloud of the population towards the danger zone. And if you're near to the edge, then you'll go over the edge first. So if you have some disease or you're born with an unfortunate set of ion channels, you might be at higher risk than other people. So what you're asking is, can you make studies of specific populations or subpopulations with a disease burden? Yes, I think we can, but it's very difficult to know the exact distributions of ion channels in the population for those subpopulations. Theoretically, it is possible.
Okay, thanks.
We have a chat. I can't see the chat myself.
I will actually read it for you. So there is a next question. So does this mean that we don't need actually the sodium peak and late, or that it does not help the prediction?
Sorry, can you read that again?
Does this mean we don't need the sodium 1.5 peak and the late sodium, or that it does not help the prediction?
Oh, right. Late sodium, I didn't talk about late sodium. Late sodium does come into play for certain drugs like ranolazine. So we can expand this method to include 3 ion channels, one of being late sodium. But for this particular drug data set we have, it only changes things by about 1%. I think there's only one drug in that whole set that matters. But I strongly feel that as we get better and bigger drug data sets, that late sodium will start playing a bigger role.